When you think of origami, you usually think of the traditional Japanese art of paper folding, taking a flat piece of paper and transforming it into some complex and imaginative 3D shape, as seen in this video. However, the same concept can be applied to complex nanogenetic sequences of nucleic acids, such as in this video. We call this DNA origami. You are taking these long single strands of DNA, seen in yellow, and folding them on top of each other. The smaller blue strands act as staples and use complementary base pairing to secure the desired sections together, just as their name implies. This folding ultimately allows you to create strong, interconnected sections that can combine with each other in various ways, making complex three-dimensional structures in virtually any form or shape. Due to its versatility, there are many present and possible future applications for this technology. I recently sat down with Dr. Key of the Key Lab for Biomolecular Nanoengineering at Georgia Tech to help us explain exactly how the folding process works and how it can be used to create nanostructures for various uses. So I'm sitting here with Dr. Key. So for someone that doesn't maybe have much of a scientific background, how would you describe DNA origami maybe in more layman's terms?
So the idea of DNA origami is to use molecules, or in this case particularly biomolecules, and design them in a specific way to enable them to self-assemble into structure at molecular level. So it's much like a molecular Lego. There's different terms, different technology in the field, DNA nanotechnology. DNA origami is one of the technique or one of the strategy people use to design DNA structures. There's other DNA design method or strategies such as DNA tiles or DNA bricks. There's different design strategies. But DNA origami is one of the most powerful or most famous technology. But no matter what, this different technology work in a similar way. The detail might be slightly different. The basic idea is you take a DNA as a building block like a Lego. You design them in a specific way to interact with each other. So you have a large number of DNA molecule or DNA strands in solution, and they are specifically engineered and allow the different molecules to interact in a way that they can assemble into a pre-designed or prescribed shape.
Okay.
So that's the basic idea of DNA origami. For example, if I design a DNA strand that interact with neighbor in a specific way, we can allow them only assemble in a specific way to assemble into final geometry. We design the DNA sequence to carry the final structure information. Plates have 96 wells, and each well has a DNA. So for example, if I design a DNA origami structure that contains 200 strands, I order maybe 2 or 3 plates. And once the plates come in, I simply mix all the strands together at certain ratio. When we put the tube in here, we run a specific program that basically raise the temperature, denature everything, and gradually cool it down to room temperature. Then everything starts to assemble in that tube, even though we cannot see it, but they assemble. After staining the DNA band with fluorescence dye, you're able to see your structure inside of gel, which form a really tight band on the gel.
Okay.
Then we can extract that DNA structure from the band. Then we do some atomic force microscope, or in some cases we do electron microscope imaging.
So what are the applications that you're currently working on for DNA origami in your laboratory?
Okay, that's a really good question. So particularly for us, there's I think there are 3 important directions right now. But the first direction is actually still basic study of DNA self-assembly. So how we can make structure larger, more complex, and more dynamic. So we can control the structure. One research focus in our lab in terms of DNA assembly is how to make the dynamic structure which can move and can change conformation after formation, so we can make molecular device, molecular machines that can perform sophisticated functions. Okay. So that's one research focus. The other direction is using DNA structures for biomedical related applications. Currently, we are focusing on using DNA nanostructures for targeted drug delivery. Specifically, focus on using DNA to deliver gene or deliver siRNA, short RNA or short DNAs to regulate cellular cell behavior and will suppress expression of certain protein or trigger a cell to have different response. For example, to trigger cell to die. The second big direction using DNA Origami in our lab is using DNA Origami for actually materials related research. In terms of material research, there's two important things. Decide what property a material has. One is the component, what molecule you have in the material. Secondly is the structure information. How do you organize those molecules inside the material? So in terms of the structure formation, a DNA molecule is fantastic. We can make all kinds of shapes, all kinds of structures with different size. However, the basic property of DNA as an organic molecule is really limited. So one idea many people in our field do this is to combine DNA with other functional materials like magnetic particles or gold particle, silver particle, which have really great optical property. So we combine those two or those materials together. So you use DNA to organize, for example, gold particle, gold nanorods, silver particle, quantum dot, magnetic particles. By doing that, we can generate a structure, have well-defined structure information, but also have this functional molecule or functional particles inside. So we can generate the structure with unique magnetic property, with unique optical property, with unique catalytic property, and those can be used for different applications. So material research is also one of the focus in our lab.
On DNA nanotechnology, so it's using DNA as a structural material rather than a genetic material. So for example, what I personally am working on is using these DNA structures to organize magnetic nanoparticles. So these can be used for MRI contrast agents, to be used in the clinic, and people have shown that if you get these particles to stick together, they work better as contrast agents. So we're trying to use these DNA structures to organize these particles in a controlled manner to get them to stick together, to really kind of understand exactly how and why you see that improvement in the contrast generation. The end goal of hopefully developing better contrast agents using this technology.
So where do you see this technology kind of being in the next 15 to 20 years or even beyond that?
That's really a hard question to answer. In general, there's two different directions for the field. One is more fundamental research, DNA assembly, and also DNA machine, DNA devices. The second direction, of course, is applications. And the field has, I think the field is about 30 years old right now. It's really close to generate some important applications. Most people in the field are putting most of their effort in the application part right now. So because of that, I'm not saying the basic research part is not important. I think that's still really interesting and still fundamental and still really important for developing the field. I feel that in next 10 years, the most important thing going to happen for the field is application part.
So now that I've introduced the basics of DNA origami and how it's made, my colleague Craig Gordon will be discussing some of its applications. Please welcome Craig to the stage.
One of the more common applications being developed right now is using DNA nanostructures as a delivery mechanism for drugs such as those used to treat cancer. Rather than me try to explain this use, however, I'll allow Dr. Dana Anderson to describe the drug delivery application from her lab.
The application for DNA origami that we are focusing on now is drug delivery. A big problem in drug delivery that researchers are trying to overcome is the body's natural aversion and defense response to foreign and sometimes toxic materials. A solution to this biocompatibility problem is using something the body already interacts with on a daily basis that is already found in the nucleus of all cells. This allows cellular and nuclear uptake much easier, delivering the drugs to where they need to go. DNA is amazing in its complexity to fold into virtually any shape and not be attacked by the natural defenses of the immune system. How we do do it is this. We take long strands of viral DNA and program to self-assemble into triangular tube structures with the help of smaller, staple strands. The anti-cancer drug doxorubicin is non-covalently intercalated with the DNA tubes with high loading efficiency. The nanostructures are taken in by the cells, and the biosynthetic inhibitory effects of the dox causes apoptosis to cancer cells. However, the next set of problems that still need to be addressed are targeting cancer cells specifically and combating multi-drug resistance.
It is very interesting to see the forefront of technology and understand the scientific developments of DNA origami to be used in the future. Now, let's hear from Mrs. Origami, who will show us how DNA technology is currently being used.
Hi, my name is Ori, and I'm the founder and CEO of Gatapont GmbH. Our company was founded in September 2014 in Braunschweig, Germany. We're a spin-off of the Technical University of Braunschweig from a lab focused on the field of super resolution imaging. So we've taken this from research to industry. We focus on creating probes for fast, easy, and precise quantification of super resolution systems. These probes allow you to check the resolution of your microscope with the precision of a few nanometers. In order to obtain this level of precision, we use special nanostructures called DNA origami as breadboards. The breadboards are used for placing single die molecules in an exactly defined pattern. The products are individually designed according to our customers' wishes.
Alright, now let's welcome back Anna to the stage.
Thanks everyone for listening to our talk on DNA origami. We're glad you could join us. On your way out, don't forget to fold up those chairs and pair up with your partners.
Well, Anna, I think that's enough DNA talk for today. Good night, folks.